All right, part 3. I lost track of my 15-minute window and got cut off in the last one. We're now doing part 3 of the population genetics lecture, and that was actually decent timing because I needed to think about what I was saying for a second. So one of the implications of Hardy-Weinberg is that if we know allele frequencies, we can calculate genotype frequencies. So we can always calculate allele frequencies from genotype frequencies. So we can always go from genotype to alleles. Genotype, let's say, whoops, frequency, and calculate the allele frequency, because that's a given. But we can only calculate allele, hold on, let me write this. Allele frequency to, we can only go from allele frequency and use that to calculate genotype frequency if we're in Hardy-Weinberg equilibrium. So that's one of the implications of Hardy-Weinberg equilibrium, and the other one is that population doesn't evolve. And then another one, which I actually forgot to mention in class, was that it really only takes one generation of random mating to restore Hardy-Weinberg equilibrium. So Hardy-Weinberg is really about these genotype frequencies. Hardy-Weinberg is only about genotype frequencies. It says less about allelic frequencies. Okay. So if we want to know if one particular locus in our population is under Hardy-Weinberg equilibrium, there's a certain approach that we can use. Let's say we have these observed genotypes. We can calculate P and Q from these, like we did before. And then from these P and Q, we can calculate the P squared, 2PQ, and Q squared, which is what we expect under Hardy-Weinberg equilibrium. Okay, expected frequencies under Hardy-Weinberg equilibrium. Then we want to know if these frequencies, which is what we expect, matches these frequencies, which is what we observe, and we use a chi-square test to do that. Okay. So we can also use Hardy-Weinberg to calculate allele frequencies given phenotypes. So far we've really only been talking about genotypes and allele frequencies. Well, what about phenotypes? Let's say we have pea plants and tall is dominant over short. We have a certain number of individuals that are tall and a certain number of individuals that are short, and we want to know what's the frequency of the short allele in this population. So let's say we have tall, and if we say have big T and little t alleles in this population, tall plants could be big T, or they could be big T, little t. Short plants could only be little t. Now we have 19, so our frequency of little t in our population is 19 divided by 100 equals 0.19. That's our frequency. So from this, how do we get Q? Well, Q is basically going to be, since this equals, and we're assuming that we're under Hardy-Weinberg equilibrium, like we say here, is actually Q squared, then Q is the, I don't know how to do this on here, square root of 0.19, whatever that is. Now, how do we get P then? Well, P plus Q equals 1. So then we just subtract this from 1, and then you get P. So that's how that's done. Like so. Okay, so that's Hardy-Weinberg, and now we get to talk about what kinds of things cause deviation from Hardy-Weinberg. All right, so one of these things is non-random mating. If we go back up to our, where are they? Our assumptions, one of the things that we assume is random mating. So there are two types of non-random mating. We can have positive assortative mating, and we can have negative assortative mating. So positive assortative mating is when individuals who mate are more similar. Individuals, well, let's say similar individuals are more likely to mate. There can also be negative assortative mating, where dissimilar individuals are more likely to mate. One example of this can be found, go to a mall where there's a crowd and look at the couples. They're going to often look similar. Humans often mate by height, weight, race, maybe geographic location. Okay, for example, negative assortative mating has been found in mice and humans at the MHC locus. So individuals with a certain genotype at MHC find individuals with a different genotype more attractive. So let's think about what this means. MHC is the major histocompatibility complex. We learned about it in the immunogenetics lecture. And it is involved in immune response. And there are multiple loci in the MHC, each with many, many, many alleles. And so there are many variations of genotypes for MHC. We tend to be more attracted to, and this was the Smelly t-shirt study, where women were given t-shirts to smell, and they had to rate them based on, I guess, whether they smelled good to them. So these were men's t-shirts that they wore for, I don't know how long, and women had to smell them. And people did this also with mice, and I think the mice had to smell urine. So I guess, given the choice, I would take a t-shirt. So women were more likely to rate a t-shirt belonging to a man that was worn by a man who had a dissimilar MHC genotype than hers. And if you think about why this would happen, MHC is a locus where it's beneficial to have a lot of variation, and it's beneficial to be diverse because then you have greater flexibility in dealing with different antigens. So that's the evolutionary just so story for that result. One example of negative assortative mating. Okay. All right. So under positive assortative mating and negative assortative mating, the alleles that are the loci that are most affected where their allele frequencies are going to be changing because of this are only the ones that are directly related to the traits that are being selected by mates. So in the MHC, it would just be MHC, and under, say, height, it would just be the genes that are associated with height. A special case of positive assortative mating is inbreeding. And under inbreeding, this is when mating between related individuals happens. And unlike these cases where just a specific subset of loci are affected, inbreeding affects the whole genome. The opposite of inbreeding is outcrossing. It can also be called outbreeding. And this is mating between unrelated individuals. And one way to think about this is communities that tend to marry within their own community, they tend to be more inbred. And I'm not saying that they're playing banjos and some of them do have health issues, but it just means that on a population scale, they have a higher what we call inbreeding coefficient. Coefficient, which is the probability that two alleles are identical by descent. And now, what does that mean? Okay, descent or IBD. Okay, so inbreeding coefficient is typically designated as F, named after Fisher, of Sir Ronald Fisher, who was a very, very famous population geneticist. And so on a population level, communities that tend to marry within their community have a higher than usual inbreeding coefficient. Whereas communities that tend to marry outside of their community are what we would call outcrossing. Okay, so inbreeding, what do I mean by identical by descent? All right, so what this means is that given a pedigree in which inbreeding occurs, down here we have our inbred individual. This is our only inbred individual in this pedigree. We're all good up until this guy. So he happens to be homozygous at this particular locus, and it also happens to be that both of these alleles originated in a single ancestor. So that's what we mean by identical by descent, whereas both alleles are descended from the same copy in a common ancestor. So here, this, great-grandma gave this A1 allele to both of her daughters, who then passed it down to a daughter, and then to a son, and then to this individual, and then it was also passed down on the other side of the pedigree, and they wound up the same, basically. And so the inbreeding coefficient is the probability in a given population that an individual has two alleles that are identical by descent. Okay. So that's on the other hand. If alleles are not identical by descent, they can be identical by state, where both of these individuals are homozygous for this A1 allele, but in the previous slide, that individual's alleles both originated in one person. Whereas here, both of each of these alleles originated in a different person. Okay, so this is an outbred or not inbred pedigree. So we're A-okay here.